Hello everyone. In this practical class, we'll try to learn how to estimate hemoglobin in the lab. Aim is to estimate hemoglobin by Sal's method. So principle here is hemoglobin is converted into stable acid hematin by addition of n by 10 HCl. This concentrated solution is diluted with water till its color density matches with that of standard. The apparatus that we require are Salis comparator, Salis tube, that is also called as hemoglobin tube, Salis pipette, which is also called as hemoglobin pipette, a thin glass rod stirrer, N by 10 HCl, distilled water, spirit swab, and lancet. So this is Salis hemoglobinometer. So this is color comparator, and this is hemoglobinometer tube, and this is hemoglobinometer pipette, and this is the stirrer. Okay. So Salis color comparator is made up of uh, the tinted glasses which are colored. So then the hemoglobinometer, it is marked up to the mark of 2, and where you will be taking the blood, and HCl is going to be mixed, and this is hemoglobin meter pipette, where you are going to suck in up to the mark of 20 microliter mark. Okay. And this is the stirrer. So this is another diagram of Sally's mm, hemoglobinometer. Uh, then this is the pipette. This is the n by 10 HCl dropper. Procedure is take a clean and dry Sally's tube and place it in the comparator. With the help of dropper, add n by 10 HCl up to the mark of 2 gram on the percentage scale. Under aseptic precaution, then prick the finger. Wipe the first 1 or 2 drops of the blood. Obtain a good size drop of blood. So then draw the blood up to the mark of zero point 0.02 ml or 20 cubic millimeter mark. Avoid air bubbles. If present, discard and repeat. Dip the pipette immediately in Sally's tube containing HCl and gently blow uh, till the blood is expelled. Then rinse the pipette twice or thrice with HCl. Mix the contents gently by stirring with a glass rod. Place the tube in the comparator and wait for 10 minutes. During this period, the RBCs rupture and hemoglobin is liberated, reacts with 1 by 10 HCl to form the stable acid hematin. Go on adding distilled water drop by drop and mix it till the color matches with that of the standard tinted glass against the natural source of light. When the color of the solution matches, note down the lower meniscus reading and express the hemoglobin content as so many grams per 100 ml of blood or deciliter of blood. So then calculation part, we can calculate the oxygen carrying capacity of the blood. So that will be hemoglobin in gram percent into 1.34, because 1 gram of hemoglobin can carry 1.34 ml of oxygen. And note that 1 gram of hemoglobin carries 1.34 ml of oxygen, and note that 1 gram of hemoglobin carries 3.34 milligrams of iron. Okay. So then it will be reported like the hemoglobin content of my blood is so many grams per 100 ml or deciliter. Okay. And oxygen carrying capacity is expressed in terms of so many ml per 100 ml of blood, and the iron content is expressed in terms of so many milligrams per deciliter of uh, blood, or it is so many milligram percent. So normally the hemoglobin in at birth is 23 gram percent. At 1 year it comes down to 12 gram percent. In adult male it ranges from 14 to 18 gram percent, uh, with an average of 15.5 gram percent. In female, uh, the range is between 12 to 15.5 gram percent with average 14 gram percent. So this hemoglobin can also be utilized to clinically differentiate the anemia into mild, moderate, and severe anemia. If the hemoglobin content is less than 12 milligram percent, it is called as mild anemia, and if it is less than 9 gram percent, then it is called as moderate anemia. And if the hemoglobin percentage is less than 6 gram percent, so then it is called as severe anemia. Thank you.